insulin. Insulin is a hormone that's secreted from the beta cells of the pancreas. It is stimulated to be released with an increased production in blood glucose, which is most often just from us consuming food. The insulin has two main actions. First action, it works on the liver. It works to decrease the liver's glucose output by increasing glycogenesis synthesis, or the mechanism of glucose storage within the liver. It can also increase lipogenesis, which is the body using its free fatty acids and converting that to triglycerides. Insulin is also promoting the tissues and the muscles and the adipose tissue to increase its uptake of glucose from the bloodstream. It can then store these as amino acids in the tissues, and then when it is needed, it can be released to the liver to release glucose or produce glucose. Insulin, as you can see from this graph, is released in two phases. So once you consume food, the first insulin burst, shown by this first phase here, is released within the first 5 to 10 minutes, and this is in order to suppress that hepatic glucose production. This mediates glucose disposal also in the adipose tissue and seeks to minimize hyperglycemia or elevated blood sugar during your meals and directly right after you finish eating. The second phase is released much more slowly. It's a gradual increase in insulin secretion over an hour to an hour and a half to 2 hours after you've consumed your meal. And this stimulates glucose uptake by the peripheral tissues. So this is where about 80 to almost 85% of the glucose metabolism occurs within the muscle, is during the second release of the insulin phase. The slower release of insulin during that phase allows for the body to respond to the new glucose that's been entering through the digestion system while trying to maintain a basal glucose level, or a steady glucose level, rather. As you can see from this graph, which we'll get into in a little bit, this insulin secretion is significantly decreased in patients with type 2 diabetes, and similarly to type 1 diabetes, once type 2 diabetes progresses, we have a very minimal, if at all, any insulin release. As you can see from these graphs, this is showing you insulin in relation to glucose in terms of your plasma and what is being released into your bloodstream. So you can see as your basal glucose rate is steady, once you eat, that glucose is actually going to increase because that is what's in your actual food. As your glucose increases, so does your insulin. And you can see these spikes between meals, so say for dinner, you can see the increase in your glucose due to your food that you're intaking, as well as an increase in the insulin to compensate for that increase in glucose. Amylin is another hormone that is secreted from the pancreas alongside insulin. It is produced in response to eating or nutrition, similar to insulin. This works by inhibiting your glucagon secretion, which is secreted also by your pancreas or your alpha cells. Glucagon, as we'll talk about in a minute, kind of is the opposite of insulin, so they work antagonistically. The amylin helps to delay gastric emptying, which slows down your GI tract in order to allow the foods to be absorbed and for other hormones to take effect to regulate that insulin release. This amylin also works in the central nervous system, or in the brain, to give you a feeling of satiety or a feeling of satisfaction. So what that does is that prevents you from being hungry, and makes you feel as if you're full, gives you that full type feeling. We currently have medications that mimic amylin and others that help to promote its release. So thinking about this when you're talking about medications will be helpful to you in understanding how those medications act. So as we were just talking about glucagon, this is also secreted by your pancreatic cells, but it is secreted by the alpha cells versus the beta cells where the insulin and amylin are secreted from. This is produced during your hypoglycemic condition. So this is kind of your mechanism as if your blood sugar is too low. This is your body's natural mechanism for compensating for that and bringing your glucose up to the levels that are needed for your body to function. So it elevates the blood glucose production by the liver. So as you may remember, we were talking about when we have too much excess glucose, our body turns on glycogenesis, and it produces glucose into a molecule that can be stored by our liver. This glucagon kind of switches that back the other way. So it takes this out of storage and releases that into glucose that our body can readily use. So this is opposing our action of our insulin, and it's stimulating our glycogenolysis and our gluconeogenesis, which is the production of glucose from those free fatty acids that are stored within your peripheral tissues. This diagram helps to illustrate what we just talked about in terms of what is released from the pancreas, insulin, and glucagon, and how these two kind of work antagonistically. And in terms of, you could think of it in terms of a feedback loop. When your glucose is high, insulin kicks in to decrease our blood glucose levels, and if our blood glucose levels are too low, glucagon is released in order to raise that. And it does so by working on the liver by promoting gluconeogenesis and glycogenolysis, as you can see in this diagram. So let's say that our blood sugar is really low. This stimulates our pancreatic alpha cells to secrete glucagon. This is going to promote the production of glucose from the stored glycogen within the liver, which is going to result in raised blood sugar. This also might stimulate your peripheral tissues to release free fatty acids that will also come into the liver that will be converted into glucose and released into the bloodstream. If our blood sugar is too high, this is going to promote our pancreatic beta cells to secrete insulin. Our beta cells will also secrete amylin as well, along with insulin. This is going to stimulate the glucose to be reuptake, will be taken up from the glucose into the peripheral muscle and adipose tissue in order to be stored. It's also going to stimulate our liver to halt the glucose production and produce more glycogen in order for it to be stored by the liver, therefore decreasing how much sugar or blood glucose is circulating within the bloodstream. This is taken directly from Dr. Malhotra's notes to illustrate the complicated role of the liver and how the pancreas and the liver interplay and work together. This is a great example of what happens when glucose levels start to drop during a fasting state. This, in other words, is a good example of what occurs when your blood glucose is low or in a hypoglycemia type episode. This is sort of a compensatory mechanism. So this insulin release is inhibited, as we can see by this component of the diagram. Our plasma glucose is low, so that signals to our beta cells of the pancreas to decrease our insulin production. This is going to prompt our muscles and our adipose tissue to release its fatty acids that are going to go into the liver, which is going to promote gluconeogenesis, or increase in glucose production, which is going to therefore increase our plasma glucose. As we'll talk about later, prolonged low blood sugar will resort our body to use other methods of energy. So if we do not have enough glucose to use, those fatty acids can also be converted into ketones, which will then be used by the brain and other tissues for energy. And if you think about other physiology courses that you may have had, you may recall the term ketoacidosis, and that's similar to what is happening here, is you're getting a prolonged hypoglycemia or low glucose, so your body is not having sufficient glucose to use for its daily functions, and so it needs to find another source of energy. And generally the source of energy that is next available is your proteins, so your amino acids are able to break down by your liver into your usable form, which is the ketones that are used by the tissues. So aside from the beta cell pathway, once decreased your blood glucose is low, your alpha cells of your pancreas also stimulate the production of glucagon. Glucagon is going to enter the liver. It's also going to promote your glycogenolysis. So this is going to stimulate your glycogen that's going to be released in the form of glucogen. Glucose. And this is going to promote your increase in your plasma glucose. And once your plasma glucose has been increased to a level at which the body can function, this will then form a negative feedback loop, which will turn off the alpha cells and the production of glucagon, and then once your glucose levels have increased, it will turn back on the beta cells for increased production of insulin to maintain that good glucose homeostasis. Another factor, other than your liver and the pancreas, that help to regulate glucose levels, is what we call an incretin effect. And this is the factors of your GI system, or factors of the gut. How you can think about this is as nutrients enter your stomach and small intestines, there are hormones that are released by your GI tract to help stimulate that insulin secretion. Two of these major, what we call incretin hormones, are GLP-1, or glucagon-like peptide 1, and GIP, which is gastric inhibitory polypeptide. The GLP-1 is the hormone that has been significantly more studied, and is also one of our drug targets, so we'll spend a little bit more time focused on our GLP-1 hormone. So our GLP-1 is released from L cells in our colon and ileum of our GI tract. As I said, this is produced in response to nutrients or you eating. You find receptors for your GLP-1s in your pancreas, in your hypothalamus, and in your stomach. So GLP-1 actions are glucose dependent, so since it is dependent on your response to nutrients, there really is no action in just your normal glucose homeostasis. This is just once you've introduced an outside form of glucose, which is your food. It helps to stimulate insulin release from the pancreatic beta cells. It also helps to suppress your glucagon secretion. It slows that gastric emptying and increases that satiety. So it's working on all of those three areas. So in your pancreas, it's helping to secrete that beta cell production. In your hypothalamus, it's helping to increase your sense of fullness or satiety. And in your stomach, it's helping to slow that gastric emptying, allowing more of that second phase of insulin release, allowing it to slowly be absorbed, or glucose be absorbed into the system. The GLP-1 is also inactivated by an enzyme, DPP4, or dipeptidylpeptidase 4 enzyme. This prevents these actions, and so we have drug targets that serve as GLP-1 receptor agonists that promote this GLP-1 action, and we also have DPP4 inhibitors to prevent that breakdown of the GLP-1 peptide. This is just a diagram explaining what, helping to visualize what I just explained as the action of the GLP-1 type receptors. As you can see, you have them in your pancreas, hypothalamus, and stomach. This helps to promote insulin release, decrease your appetite,